In this video, we'll discuss about uronic acid pathway. It is an alternative oxidative pathway for glucose. Glucose will be converted into glucose 6-phosphate by the enzyme glucokinase. One phosphate group is denoted by ATP. Then glucose 6-phosphate converted into glucose 1-phosphate by phosphoglucomutase enzyme. Then this glucose 1-phosphate then converted into UDP glucose by UTP glucose pyrophosphorylase enzyme. Last, UTP glucose is converted into UDP glucuronic acid by UDPG dehydrogenase enzyme, which is NADH dependent enzyme. And this active form of glucuronic acid has various function. What are the function it have? There are used for conjugation of bilirubin, conjugation of steroid, conjugation of various drugs, and synthesis of glucose aminoglycogens. After this, UTP glucuronic acid will be converted into D-glucuronic acid by the enzyme UTP glucuronidase enzyme. Glucuronidase enzyme, 1 molecules of UTP will released. Then the D-glucuronic acid is converted into L-glucuronic acid by the enzyme glucuronate reductase. NADPH2 will be converted into NADP. This L-glucuronic acid is converted into ascorbic acid in lower animals. In lower animals, it will converted into this L-glucuronic acid converted into ascorbic acid. But this one cannot converted this ascorbic acid to human. This one cannot converted into ascorbic acid in human. In lower animals, it will converted into vitamin C ascorbic acid. In lower animal, this L-glucuronic acid first converted into L-glucuronoacetone by this enzyme gluconolactone oxidase enzyme. Then it will be converted into 2-keto L-gluconolactone. Then this 2-keto L-gluconolactone acted upon enzyme gluconolactone oxidase enzyme. It will converted into ascorbic acid. Human cannot synthesis because the absence of gluconolactone oxidase enzyme. This enzyme gluconolactone oxidase enzyme absent in human being, presented in lower animals. In human, this L-glucuronic acid is converted into 3-keto gluconate 3-keto L-gluconate by this enzyme L-gluconato oxidase. NADPH is converted into NADP is converted into NAPH2. 3-keto L-gluconolate 3-keto L-gluconolate will be converted into L-xylose by decarboxylation reaction, removal of one carbon dioxide. Then this L-xylose is converted into xylitol. L-xylose is converted into xylitol by xylitol dehydrogenase enzyme. NADPH2 is converted into NADP. Now this xylitol is converted into D-cellulose by this enzyme cellulose reductase. NAD is converted into NADH2. Now this D-cellulose is converted into D-cellulose 5-phosphate. By this, with the help of enzyme cellulose kinase enzyme, ATP is converted into ADP. Finally, this D-cellulose 5-phosphate will enter pentose phosphate pathway. So, absence of these two enzyme, any of these two enzyme, xylitol dehydrogenase, cellulose reductase, absence of any of these enzyme leads to condition name is called as pentose urea. That condition name is called pentose urea. So uronic acid pathway, it is an alternative oxidative pathway for glucose. This is important of this pathway is to provide UTP glucuronic acid, which is the active form of glucuronic acid. Then UTP glucuronic acid is used for conjugation of bilirubin, then conjugation of steroids, conjugation of various drugs, and synthesis of glucosamino glycan GAG. Then steps, first glucose is converted into glucose 6-phosphate. Glucose 6-phosphate is converted into glucose 1-phosphate. Glucose 1-phosphate is converted into UDP glucose. Then UDP glucose is converted into UDP glucuronic acid with the help of enzyme UDPG dehydrogenase. This one is NADH dependent enzyme. Then step 5, UDP glucuronic acid will converted into D-glucuronic acid, glucuronic acid. Then D-glucuronic acid is converted into L-glunic acid, glulonic acid. Then L-glulonic acid is converted into ascorbic acid in the case of animals, not converted into human. First, how this L-gluronic acid converted into ascorbic acid in animals. We will discuss L-gluronic acid first converted into L-gluronolactone. Then this L-gluronolactone will be converted into 2-keto L-glunolactone. Then it is converted into ascorbic acid, that is vitamin C. In human, L-glulonic acid is converted into 3-keto L-glulonate by the enzyme of L-gluconate oxidase. This one, NADP is converted into NADPH2. Then this 3-keto L-gluconate is converted into L-xylose. Then this L-xylose is converted into xylitol. Then this L-xylitol is converted into D-cellulose. Then D-cellulose is converted into D-cellulose 5-phosphate. Then this D-cellulose 5-phosphate finally entered to pentose phosphate pathway. These are the statement form. This L-cellulose is converted into xylitol with the help of enzyme L-xylitol dehydrogenase. When this enzyme is absent, defective, this L-xylulose cannot converted into xylitol. So increased concentration of this L-xylulose which further excreted through urine. Under this pentose urea, one among Gerard Detrat, that is inherited metabolic disorders, and among these 4 disorders, pentosuria, albuminism, cystinuria, and alkaptonuria. This essential pentosuria, inborn error of uronic acid pathway. It is benign condition and excretion L-xylulose in urine. This uronic acid pathway disorder name is called as essential pentosuria. This L, that is xylitol dehydrogenase and xylulose reductase enzyme. Any of these enzyme absent means it leads to pentosuria. L-xylulose in urine gives positive result in penetic test. In drugs like barbiturate, barbiturate and aminopyrine, etc., will induce the pathway and increase this xylose urea. This condition does not produce any harm.